Good morning, everyone. How are you guys doing? I'd like to welcome you all to the third day of the FDA MHRA Health Canada Joint Symposium. It is my great pleasure to kick off the bioequivalence session today, and I highlight some of the work that we have been doing in the areas of bioavailability and bioequivalence. Today's presentations are continuation of the discussions on important GCP topics that were discussed in the past 2 days of the symposium. However, this morning's focus will be on a specific GCP area, which is for bioequivalence studies. And that is slightly different than the conventional efficacy trial that we've heard for the past days. As you can see, this is a joint symposium with the MHRA and Health Canada and FDA. And as such, what we aim to achieve from this symposium is to share with you the collective knowledge and experiences from these agencies on inspections and regulatory oversight on bioequivalence studies, and to convey harmonized message with the hope that it helps you guys to do a facilitated drug development, global drug development. The international collaboration is one of the important strategic goals of our office, the Office of Study Integrity and Surveillance, as well as for the FDA overall. The OSIS Bioequivalence International Collaboration, specifically with the MHRA, has started more than 8 years ago. And as you can see from this symposium, the collaboration now has expanded to include Health Canada, which we started off years ago. This international collaboration has been very successful because we all work together with a common goal of making sure that the regulatory data submitted to our respective agencies are robust and reliable, and the safety of subject and participant who goes into the clinical trials are well protected. As part of the collaboration, we conduct joint or observational inspections, exchange information about the inspection details and outcomes, clarify expectations to industry on study conduct when it's necessary, and share any tools that might help the regulatory oversight. Again, by working together, we hope these efforts convey a harmonized message to industry on expectations on study conduct of bioequivalence studies across the agencies, and help us streamline the planning and execution of your own studies. As for the topics of the presentation this morning during the bioequivalence session, we have 3 very exciting sessions planned. In the first session, we will present an overview on our experience on regulatory remote evaluations. When COVID-19 hit the world at the beginning of 2020, and essentially every aspect of our lives was impacted by that, our office in FDA led an innovation and did a really groundbreaking work, which was to develop a remote evaluation approach. And it was these efforts that allowed us to continue our work and to accomplish FDA's mission of protecting public health throughout the pandemic. Among other things, we crafted a communication plan with the industry, made sure that the remote approach that we are developing is as flexible as possible given the very fluid at the time, and made the very best use of all the technologies and communication platform available, which, by the way, weren't that much at the time, as you know. Yet, we tried to make sure that we maximize the efficiency and the evaluation capability so that we can make adequate review of the study conduct and study information and make robust conclusions about the reliability of study data. Initially, we called this remote tool as a remote regulatory review, in short, RRR. Then in April 2021, FDA issued a guidance for industry entitled Remote Interactive Evaluation, RIE, for Drug Manufacturing and Bioresearch Monitoring Facility during COVID-19 public health emergency. And that was the first public announcement that explained FDA use of a remote tool. Since then, the agency recognized the need for formalizing a procedure for remote evaluation across a variety of compliance programs. In July 2022, FDA issued a draft guidance titled Conducting Remote Regulatory Assessment Question and Answers. And in there, it also explained that the terminology, the Remote Regulatory Assessment, RRA, that you heard yesterday, is a category of activities that FDA uses for remote evaluation. And it encompasses the remote regulatory review and the remote interactive evaluation as types of RRAs. And as some of you may know, just a few weeks ago, FDA issued an updated draft guidance, which is now open for public comment for the 60-day comment period. In parallel to FDA's efforts, our colleagues in MHRA also developed a remote inspection paradigm. And through the collaboration that I mentioned earlier among MHRA, Health Canada, and FDA, we exchanged the tools and approaches, advantages and challenges, and notes on similarities and differences in how we conduct remote evaluations. It has been extremely valuable to learn from each other, and now I'm very excited to see how the two presentations this morning, one each from MHRA and the FDA, in the first session will inform you about the two agencies' remote approaches and provide you with the important take-home messages so that it helps you to get prepared for your future engagement and inspections by these agencies. So the next two sessions that follow, the RRA presentations, will touch on the actual bioanalytical and clinical aspects of bioequivalence studies and the perspectives from the regulatory inspections. Based on the current business models and industry trend, we see that it is generally the case that the sites that conduct the clinical phase of bioequivalence studies and those that test and analyze the biological samples from study participants are separate entities. And depending on the business relationship between those two sites, there could be more or less of communication between those sites about study design, sample collections, sample transfer logistics, and etc. And in some cases, there could be additional sites in addition to the bioanalytical and clinical sites that play various roles and functions during the study conduct. And as more entities get involved, it could easily get complicated when one tries to reconstruct all the study activities and to verify the integrity of study samples. And if there happened to be an information gap, there could be a delay in application review or even approvals. So given the pivotal nature of the bioequivalence study in generic drug approvals, you can appreciate how important it is for us to verify if subjects receive the right sequence of drug treatment and the drug concentration data submitted to agency are reliable. So for these reasons, FDA, MHRA, and Health Canada conducts inspections at both clinical facilities and bioanalytical laboratories. As for the clinical phase, we expect the bioequivalence studies are conducted per GCP principles, and documentation is complete and contemporaneous. Studies should be conducted per protocol, and all deviations should be reported. Importantly, the rights and welfare of study subject or study participant should be appropriately protected. For the bioanalytical phase, we want to ensure that the method that was used to analyze the study samples is fully validated and works robustly during sample analysis. The tools and the processes that measure drug concentration should be clearly established and properly controlled. Throughout all the phase of the study, from the subject screening and enrollment to sample analysis to data reporting, we expect that a robust quality management system be in place. So I know many of you working in the pharmaceutical field put the patient in the center of everything you do and try to do the right thing. And we and the public really appreciate that. In some cases, however, we also have seen some concerns with data from study sites. I feel that we as regulators need to let the industry know of our expectations on study conduct and best practices and pitfalls we have seen. And that is exactly why we are holding a conference like this today. We perform a number of bioequivalence inspections each year, and the presentations from the 4 speakers in the 2 sessions this morning will highlight some of the common or unique observations from our inspections that are worth noting, so that you can pay attention to those aspects when you're conducting your own study. So we have a morning that is full of important and informative presentation, as well as opportunities for you to ask questions and discuss with the esteemed panelists. I would like to express my sincere appreciation to all the speakers and moderators who worked so hard to make today's event informative and impactful. I hope that you are as excited as I am, and I look forward to having engaging interactions and lots of great discussions. Thank you so much. So I'd like to hand off the podium to the moderator of the first session, Dr. Seon Kasim.